Very warm welcome to another one of our webinars series brought to you by Dot. My name is James Logan. I'm the CEO and one of the founders of Dot. Now, before we get started on today's seminar, over the coming months, we're going to be having further seminars from different members of our expert team who are going to be covering a whole range of fascinating topics. And I just wanted to highlight a couple of the other webinars that are coming up in the next couple of weeks, just so that you know about that as well before we get started with today's seminar. So on Monday next week, put this in your diary, Dr. Frederick Seelig, former partnerships manager for the Global Vector Hub, is going to be delivering a webinar on the Global Vector Hub, which is essentially an open access interactive platform developed by Dot that has the potential to revolutionize how we prepare for and respond to vector-borne disease outbreaks. So that is going to be a really good one to come along to. And then on Wednesday, the 22nd of April, Rory Fry, who's our sales director for our Bug Sense product, which is our bedbug product, he's going to be delivering a webinar highlighting the key pest control challenges that are impacting the hospitality industry and what can be done to solve the pest problem. So a lot of us probably spend time in hotels. The last thing we want to do is share our hotel room with bed bugs or take those home. So do come along to that webinar as well, which proves to be, I'm sure, really, really interesting. A little bit about the format of each webinar. So essentially we have a short talk, usually about 20 to 30 minutes, followed by a Q&A session. So please can I encourage you to post any questions that you have for Stephen in the chat, and I will field those questions at the end. So we can't turn our mics on, but we can post them in the chat, type them in the chat, and we'll see them there. Thank you very much. Now on to today and why we're all here. So I am delighted to introduce Dr. Stephen Bryden as your speaker for today's webinar. The webinar is entitled On the Menu, What Really Happens When a Mosquito Bites and Why It Matters. And I've seen a sneak preview of this talk and it is going to be very interesting. A lot of the information in this blew my mind. Stephen is a scientific business development manager here at Dot, working under the ArcTech Innovation, which is essentially our contract research organization, services part of the business. Stephen is a hugely dedicated and enthusiastic scientist, which you'll hear in a minute. He's got a real passion for translating medicine and neglected tropical diseases. His PhD was focused on key aspects of the innate immune response to arboviruses at mosquito bite sites, which is exactly what we're going to learn about very shortly. So without further ado, I shall pass over to you, Stephen.
Thank you very much. I'm just going to quickly share my screen. It's worked earlier on, so hopefully all good. There we go. Perfect. Perfect. Thank you so much, James. Thanks for the introduction. And thanks to everyone who's listening, either live or on the recording, which I'm sure we'll be able to get the recording later on. It means a lot that people take the time out of their business and their busy schedules to tune into these webinars. So thank you again. It's really an honor to present some of this in this series and to share some of the knowledge that I have and what I believe is a really critical but ultimately underappreciated aspect of mosquito-borne disease, and that is the bite site, the mosquito bite site. So let's get into it. So as James said, this is a year-long webinar series, so we've got lots that we'll hear about in terms of what makes us attractive to mosquitoes or how mosquitoes navigate their surroundings and their environment, and to find a host to get their all-important blood meal. But today I want to focus in on this very small, very short, but extremely consequential moment, which is the actual moment when the mosquito bites. So this is something we've probably all experienced at some point in our lives, and if we haven't, you're very lucky. It's a very short experience, usually lasts less than a minute. But this short encounter can either leave you with a bit of an itch, or it can leave you with devastating and profoundly devastating impacts on our health and our livelihoods. So I want to take a look at how all this works, what it means for disease and disease progression, and then ultimately what it means for the future of after-bite care. So first I want to start off with is a bit about the mechanism. So I think there's a lot of a misconception that a mosquito is a flying hypodermic needle or is a straw that goes in and just sucks up blood. But the mosquito proboscis, the mouth parts, is a very highly engineered and a highly specific tool with lots of different elements to it. So underneath this sheath, you've got these different components. You've got mandibles, which pierce the skin. And then inside the mandibles, you've got a set of maxilla. So these got a serrated edge and they effectively saw through your skin and then widen the wound and hold the wound open. And that allows the other mouth parts to go in search for the mosquito's lunch. So it goes in, it's got this hyperpharynx, which is used to deposit saliva and spit into the skin, and then the labrum, which is used to bring up the blood and take the meal itself. So once inside, I'll put this lovely video. These videos are absolutely fantastic. They're from a great paper from 2012, and it just really brilliantly highlights the different components of the mosquito proboscis. As you can see there, it's not just the one straw that goes in, the one needle. There's lots of different elements. So I'm going to use a little bit more of that video to explain a little bit about how you're finding your target. So once you're in the probing mouth parts of a mosquito, hunt for blood vessels. So it uses chemical receptors to try and get it towards its lunch. And as you can see, it's quite a lot of movement. There's quite a lot going through a lot of the skin. All the while it's doing this, it's spitting, causing a little bit of local damage as well. And then eventually what it's trying to do, if we can wait to see, is find a blood vessel and then start cannulating. There we go. And having its blood meal. So what I want to highlight here is this process takes quite a while. I mean, comparatively speaking, it didn't just go in and go straight for the blood vessel. It took a little bit of time to find it. And all the while it's doing that, it's spitting saliva into your skin and then eventually into the blood vessel as well. So just a little bit on saliva and what it is and what it, and why it's important. Saliva in mosquito is an incredibly pharmacologically active and sophisticated substance. It needs to perform a lot of different tasks. So you've got anticoagulant molecules in the saliva that prevents our blood from clotting and allows the mosquito to take a meal. We have molecules which contribute to vasodilation, so increasing the size of the blood vessel, bringing more blood in so it can feed on that blood. And then other things are antihemostatic, so preventing our natural blood bleeding control. And then, there's a whole list of other different proteins and activators in mosquito saliva. But one thing I just want to highlight is there's also quite a lot of bacteria. So often not pathogenic bacteria, but it's still bacteria. If you were to get saliva and put it onto an agar plate, it lights up. It's quite stark. And then there's also the potential to have things that we kind of are aware of, the nasty things. Things like parasites, the plasmodium that we know of from malaria, or potentially viruses as well. So there's a lot of reasons here why mosquito bites are uncomfortable and why we don't like them and why we find them uncomfortable. But one of the things I want to talk about is that itch sensation that we get. So the reaction that we probably most associate with getting a bite is just a little bit of an itch, a little wheal like this developing, and then as it goes on, getting itchier and itchier. In very extreme cases, people have more systemic reactions. They can get anaphylaxis, for example. But for the most part, it's this that we also associate with it. So a bite can be broken down pretty much, or a reaction to a bite can be broken down two phases. This immediate phase, so that's when you get this here, this 2 to 4 millimeter diameter wheal forming. It's itchy, but it's not as bad as what comes next, which is that delayed reaction where you get a more inflamed, kind of red and extremely itchy pruritic papule. So what causes the itch? It's primarily histamine. So there's a lot of different components in saliva. We have leukotrienes and tryptase, but it's mainly, we think, histamine. Histamine is a natural part of our natural immune system. It helps with vasodilation to get more blood into the area and promote inflammation. It's really good for causing edema, so fluid retention in the skin. And we also have receptors on our cells in the skin, on our nerve endings, and have receptors for histamine, which causes that itch sensation. So why we itch is not entirely known. There's a good hypothesis that it's a very good evolutionary advantage to itch. So the skin, if you think of your skin, it's the barrier you have to the outside world. So you want to alert yourself anytime that there's a breach of that barrier. So that's maybe one of the reasons why we itch. And a little bit of the mechanism of how it happens. So in this area, in the bit of the skin, we have these mast cells. So these are immune cells that are in the dermis of our skin. And inside the mast cells, we have these molecular mediators, lots of little granules of different mediators, but one of them is histamine. And on the outside of these cells, we have antibodies, IgE antibodies, which can cross-link or bind to like a lock and key mechanism. They can bind to antigens, so anything foreign in the body. In this case, it could be the saliva, which triggers the binding, and then that triggers this release of histamine. which goes on to cause the itch sensation. So what can we do about it? Generally, what we do and what most afterbite care focuses on at the moment is combating this itch and the pains associated with biting and scratching. So we've got the oral and topical antihistamines. That's probably the thing you probably most likely take if you start to get a reaction like this. We also have anti-inflammatories, so hydrocortisone, for example, can be prescribed if you have a really bad reaction to a mosquito bite. We also have a lot of analgesics, so painkillers, oral or topical painkillers like ibuprofen gel that you can put onto the site to try and relieve a little bit of the pain. And then I think for the most part, if you go into a supermarket or you go into like Boots or your local pharmacy, you're going to get something that has humectants in it. So a humectant is just a molecule which can bind moisture to the skin and it gives this kind of soothing sensation. I think of aloe vera as a classic example of this. A lot of people would apply aloe vera gels to bites. And it might not necessarily target the mechanism of the immunology that's happening inside your skin, but it gives you enough of a relief to let the immune system do its job and go through it. So that's kind of where we are at the moment, but I want to bring us on to the future and what we could offer. And to do that, we need to do a little bit of virology and a little bit of immunology. So I'll just give you a really quick crash course in what I think you need to know about mosquito-borne viruses. So if you remember earlier, I said one of the components of a mosquito bite is potentially viruses. So obviously they're not in all mosquito bites, but they might be there. And these groups, arboviruses, what we call them, arthropod-borne viruses, a group of over 300, well over 300 genetically distinct viruses, which are spread by arthropods, such as mosquitoes, midges, ticks, and sandflies. So the important viruses that you may have heard of are things like dengue, chikungunya, and West Nile viruses infect humans. But there's also agricultural and livestock associated arboviruses, things like Schmallenberg or blue tongue virus, which affects ruminants and has had a major impact on food supply and agriculture. So it's not just a problem isolated to human health, is what I'm saying. So there are more infections occurring year on year with arboviruses. And this is because the geographical range of these viruses are increasing. And there's a very complex reason for this, or several very complex reasons for this. But it's really, some of them are like climate change. So as the mosquitoes' environment, as the temperate areas become warmer, mosquitoes being able to move into these regions, kind of higher latitudes into Europe, into the United States, for example. We have greater urbanization. So mosquitoes like to not fly very far to get their meal. So they go from person to person. So the higher density we have of people, the more likely people are going to get bitten. We also have land use changes, so a lot of deforestation, and then agricultural practices, which involve irrigation, for example, provide a fantastic opportunity for mosquitoes to lay their eggs, so we get more mosquitoes. And then obviously globalization. If I'm sick and I go on a plane, I can go somewhere else. I go to this new place where there's a new mosquito who just landed from a shipping container, for example, and I get bitten, and then we can start the cycle of spreading the virus around. So that's another kind of key aspect. One thing I really want to stress is we have very limited therapeutics and only a handful of vaccines currently available. And there's a couple of good reasons for that. One is just the genetic diversity of these viruses. So over 300 viruses, we can't possibly have medicine to target every single one. One of the other things is the outbreaks are usually unpredictable and sporadic. So it's really hard to be able to stockpile medicine, even if we had it, and get it to the right place at the right time, because we don't really know when that's going to be or where that's going to be. The other major issue, and I say probably the biggest issue, is a lot of these viruses, think of dengue, they will present themselves in the beginning as a very undifferentiated febrile illness, a very similar disease. You might get a headache, feel a bit feverish, but it's not until later stages of disease. For example, when you get the virus going into the joints in chikungunya and causing arthritis, or West Nile virus going into the brain and causing encephalitis. It's not until those end stages that we really can understand what disease we've got. And that's because we've got poor diagnostics. And what we really need is real-time, rapid diagnostics to make some of the medicine we have more useful. So hopefully that's just a quick crash course in arbovirology, and we can kind of get back onto the main topic, which is the bite. So I just want to go back to how this process, the mosquito biting your skin, relates directly to virus infection and disease. So I'm going to explain and try and convince you that the bite really makes the disease worse. And it does this through this process. So it's quite a lot in the slides. I will take you through it. When a mosquito bites, it triggers, we've got the saliva going in, we've got maybe a bit of virus going in. But ultimately, what it's doing is it's triggering this local inflammation in the skin. And it's mainly dedicated towards looking at the saliva. So it's kind of ignoring the virus at this stage. That's an important thing to keep in mind for later. So you have these local skin cells are called macrophages, and they are producing different chemical signals to try and recruit other cells to the area that might be required. So, for example, this molecular signal, for those who are interested, CXCL2, it's a chemotactic cytokine, and it brings in another type of cell, which is called neutrophils. And these are kind of like your immune system's first responder. So they are short-lived, they come in, they orchestrate a lot of what's going to happen next. And in this case, they bring in a specific type of cells. They've got the more chemical signals that bring in from the bloodstream more cells into the skin. And these myeloid-derived kind of macrophage-like cells, they are really permissive to virus infection. So the virus sees these come in, it's not been doing very much, and it's like lunchtime, and they all start infecting. And they start to make these almost factories of viral replication. So they start increasing the amount of virus that's replicating in these cells, and eventually the virus starts seeding back into the blood. And eventually that virus will either get picked up by another mosquito or will go to disseminate to further tissues. It may go to the brain if it's West Nile virus or the joints if it's chikungunya. But it's really this process of the immune system reacting to mosquito saliva and the mosquito bite itself is bringing in cells which are permissive to infection. So I just want to prove that with some from data. This is a mouse model using a virus called Simliki forest virus. The virus is very genetically similar or related to chikungunya. The main difference, it infects mice and it causes an encephalitis, so it goes to the brain eventually. So normally, if you were to inject this virus and you look in the blood later on, what you'll see is very little virus actually makes it to the blood. If you were to inject this virus, the same virus, same amount of virus, into a mosquito bite, so in the context of a bitten piece of skin, you see this massive increase in the amount of virus you can see in the blood. And what actually that has the impact of is end-stage disease. So it's more than just an annoying itch. It actually impacts disease. This is a survival curve. So if you look at this as 100% survival if you just inject the virus in the mice. But just the simple act of the mosquito bite itself, a process is said that lasts 1 minute and creates a 2, 3, 4 millimeter little bump on your skin. What actually that's doing is significantly reducing the survival to infection to just over 50%. This whole process that it's going through is enhancing the amount of virus, and the more virus in the skin results in the more severe disease. So hopefully I've convinced you, but just a summary for those who are just tuning in, it's just saying that the mosquito saliva acts on skin immune cells and blood vessels to recruit the cells which are really permissive to infection. The virus likes to infect these cells. You can see it's down here again. Brings in cells, virus likes to infect them. What's really important and really interesting, I think, is that the vast majority of virus in the blood derives from this skin bite site. I think there's a misconception that a mosquito goes in, finds the blood vessel, and then it starts spitting virus into the blood, and that's how we're getting infected. But the virus definitely goes into the blood at that stage, almost certainly not enough to actually seed a productive infection. What's happening is it's going into the skin and it's infecting your skin cells in that tiny little patch of skin, and then that's eventually making its way into the blood. So one thing I will hope to convince you is that more virus at this bit of the skin equals more severe disease. So I suppose the next point is, what does this mean for the potential for afterbite care if we know the skin's so important? And I want to take you through 4 quick things. One is immune modulation, so boosting our natural immunity in the skin. One is anti-inflammatories, antiviral medication, and vaccines. So without further ado, let's get into it. So this is a little bit complicated, but I'm just going to start with this left-hand panel here. So this is what happens normally. You get bitten, virus comes in, your immune system is effectively switched off, for lack of a better word. It's not paying attention to what's happening with the virus. If anything, it starts to react more to what's happening with the bites and the saliva itself. And then ultimately that results in disease, as we've shown in the previous slides. What this idea is being able to switch on the immune system at the appropriate time, kind of like a break glass in an emergency. What we want to do is get a compound in a skin cream that basically switches on or alerts the body's, so the skin cells, that there's an infection. And by doing that, you're helping the skin cells fight off the infection at this very critical early stage. And just as previously, more virus at the bite equals more severe disease, the idea here is that less virus at the bite site by stopping it means less severe disease. And a little bit more on this. So we've done a lot of work on this in the past, and this is kind of the mechanism. So our cells in our body and our skin, they have lots of different receptors. These receptors are used to detect damage, they're used to detect pathogens, and one of these sensors is called toll-like receptor 7. It basically, its job is to look for viral RNA, so virus genome, genetic material. And similar to other receptors, it's lock and key. Once it binds to it, it starts triggering an immune response, our antiviral immune response, to try and stop cells from getting infected. This can be manipulated in a way by using drug products. So one example is this compound called imiquimod. Imiquimod can be found in a skin cream. It's formulated a topical skin cream that is currently on the market for superficial skin conditions, things like basal cell carcinoma and actinic keratosis. So what this molecule can do is it can bind to this receptor. It basically looks like to the body a big virus infection. And that immediately, as I said, like a break glass, alerts the immune system. It triggers this receptor. You get the cascade of different downstream applications for this. And then the idea being by boosting this type 1 interferon response, which is our key antiviral response that's downstream of this, we can stop infection in the skin. So, as evidence, this is the same mouse model. It's mice, Simliki forest virus, this kind of alpha virus. It's similar to chikungunya. And then looking in the blood for virus. And then if you just, in the context, I should say, all these experiments are done in the context of a mosquito bite as well. So, as you can see, a lot of virus is being found in the blood when you just inject it, mosquito bite, sorry. But then when you inject it in mosquito bite and treat it, treat the skin 1 hour later with topical skin cream, rub the cream on, you can see the virus in 24 hours can be detected. And this is far easier shown, I think, here in this image of a brain. So this is a mouse brain infected with a virus, some leaky forest virus, which produces a red fluorescent protein. So where you see red is virus in the brain. And this is untreated. And then this is, again, treated in the skin within the first hour of infection, putting it onto the little bump that the mosquito causes. And then you can see no virus makes it to the brain. So these mice don't get the encephalitic disease. So that's the kind of the main principle of immune modulation in terms of mosquito-borne viruses. So I want to quickly move on to anti-inflammatory medication. So I want to start off by saying that we don't actually have much data on this at all. And I'd be really interested to see people in the future trying to apply this idea to see what's going to happen. So the idea here is this pathway, which enhances infection. There's so many different parts of this pathway we can interrupt or we can disrupt. And it would be interesting to see if we disrupt this, if we stop the enhancement of infection, does that stop disease? So this is an experiment that was done not therapeutically, it's not a kind of a therapeutic strategy. It was purely to work out the mechanism of these neutrophils, these pink cells. But it does kind of illustrate the point that if you interfere with something, you stop the disease. So we've got no bite here, just normal infection with virus. This left-hand column is your normal control with the bite. You see this enhancement of infection. And then you can actually deplete these neutrophils. So you can stop this process and theoretically all the downstream processes. And as you can see, massive decrease in the amount of virus found in the blood. 100% would not recommend depleting neutrophils as a therapeutic strategy. They're far too important, these cells. But it just shows that you can interrupt the immune response, the inflammatory response, which causes disease, and you can have a reduction in virus. So the question would be, what anti-inflammatory medication could we incorporate into a topical after-bite care to stop or alter the process of enhancing the infection? So I don't have an answer for you, but it's an interesting point to think about. Similarly is antiviral medication. So as I said before, we currently have very limited antiviral options. A lot of that is because, so I should state before I say that, we have the molecules, we found them in vitro, they work. They can disrupt many aspects of the virus life cycle. They can stop virus replicating, for example, but often they don't work particularly well in the clinic. And one of the reasons for that is, again, the timing. Often you get a mild, undifferentiated illness when you get an infection at the beginning. And by the time you get a more severe disease or a diagnosis, we're already at the point where these antiviral medications don't work. as well, often because the virus has already got to the tissue, the distal tissue where it causes disease, or it's triggered an inflammatory immune response that's already doing damage to the body. So it's all about timing with antivirals, and it would be interesting to see if we could use these existing antiviral molecules packaged in a vehicle which goes on the skin, like a cream or a gel. And it would be interesting to see if that alone could disrupt this last part, which is the actual replication of the virus in the skin. So obviously it's very early on, so this is a perfect opportunity. We've got maybe pan-flavivirals that we can use against dengue or Zika. If that's in a skin cream, could we stop the virus here? And that'd be really interesting to see moving forward. The last thing I want to talk about is a bit out there and actually quite exciting. And I honestly I didn't think this was going to work, but this amazing paper came out last year that showed really promising results. And that's to use a novel vaccine to target mosquito saliva. As I said earlier on, the genetic diversity of mosquito-borne viruses means it's really challenging to develop individual vaccines against lots of different viruses. So why don't we look at something that's common with them all? And that could be the saliva. So this is a recent study where they inoculated patients in a clinical trial with a vaccine against, I think it was three Anopheles saliva proteins and one Aedes saliva protein. And then they challenged the same people after a boost with a mosquito bite. Took the skin, looked at the transcriptomics of the skin, looked at what was happening in the skin, and really interesting results. What they found is that genes which are associated with antiviral activity, so these interferon stimulated genes, were all enhanced or increased, and the inflammatory genes that I showed you right at the beginning and associated with bringing in more cells, which the virus likes to infect, so those enhancing inflammatory genes, were all downregulated or decreased. So we have no data at the moment how this actually plays out during a virus infection. But it would be an interesting thought, it's an interesting thing to pose that perhaps we could prevent virus infection purely by targeting the saliva or mosquito bite itself with a vaccine. So that's one of the kind of the interesting novel aspects of the future. So hopefully in the past 20, 30 minutes, I've managed to convince you that we have a discrete location that we can use for novel therapeutics. The mosquito bite is not a very big part of our skin, comparatively. It's a couple of millimeters in diameter. And by targeting this, we can do a number of things. We could boost the natural immune response in that small bit of skin to try and prevent virus from replicating. We can disrupt the inflammatory pathways that the virus exploits to replicate itself. So remove the recruitment or prevent the recruitment of these virus factory cells to stop these kind of areas of replication happening. We can also potentially formulate antiviral drugs. This is a great area for targeting with antiviral drugs. And then lastly, we could potentially vaccinate against the mosquito saliva itself, causing an upregulation of antiviral genes and a decrease in these bad, deleterious inflammatory genes at the site itself. So this is the future, hopefully. But back to the present, I just wanted to give us a little bit of advice about what the NHS would recommend. The NHS recommends if you have a mosquito bite to apply an ice pack to reduce the swelling, taking painkillers such as ibuprofen or paracetamol if the bite is really painful. One thing is really important is do not scratch a bite. I know it's so difficult. It's some of the worst experience ever having a really itchy mosquito bite, but don't scratch it because that might not have any effect on what's happening inside your body, but it could potentially get infected with a bacterial infection, for example, if you have an open wound. So it's really important not to scratch a bite. You can manage the itch with antihistamines. And one thing is to avoid home remedies. There's lots of crazy things you'll find on blogs online. Just, if in doubt, consult a medical professional or your GP. So that's kind of after bite care. But I suppose the most important thing to take from this is what is the best protection for me now? And really, it's insect repellents first and foremost. So if you're going out in an area where you know there's going to be lots of mosquitoes, insect repellents, these ones that I've listed here, the ones we'd recommend work really well. The other thing you can do to prevent biting is just wearing protective clothing. So long sleeves, wear long trousers, try to avoid having too much exposed skin, which the mosquito can bite. So basically what I'm saying is the best way to treat a mosquito bite right now is not getting bitten in the first place. The other things we can do more as a society is work more about insecticides, trying to reduce the population of mosquitoes, and then other kind of more novel ways of reducing the population. Things like removing the breeding areas. So if you've got a lot of standing water, try and remove as much standing water in your land. If you've got alternative is GM solutions. So there's a lot of genetic modified solutions kind of cropping up, and you'll see them in the news quite a bit, trying to reduce the number of natural mosquito population. So I think that is me for today. So thank you very much. Got one last slide. Just want to do a quick plug for Digital Odor Technologies. This is our stable. We've got the parent company Dot, which is using next gen odor sensing technologies to detect pest and disease. We have our analog bedbug detection trap, the Bug Sentry Pro, which allows us with early rapid bedbug detection. And then Arctic Innovation, which is what I'm the scientific business development manager for. Arctic Innovation is our contract research organization. We do a lot of testing and validation in the lab and in the field, covering everything from public health, animal health, human health, agricultural health, and plant health, and environmental health. So all the health. So thank you very much.
Well, I think let's wrap it up there. Once again, Stephen, thank you so much for a fascinating talk. It was absolutely brilliant. And we will be sharing this recording so that other people can watch it as well. So if you'd like to share that, if anybody's here who enjoyed that and would like to share it with their network, then please do. Thank you so much to everybody for coming along. Just a quick reminder of the webinars coming up. So on Monday the 13th, that's next week, Dr. Frederick Seelig is going to be delivering a webinar on the Global Vector Hub, an open access interactive platform developed by us that has the potential to revolutionize how we prepare and respond to vector-borne diseases. And then on Wednesday, the 22nd of April, Rory Fry is going to be talking about pest control challenges that impact the hospitality industry. So all about hotels and bed bugs, essentially, and what can be done to solve that problem. So we absolutely welcome any feedback on the webinar series, and we'd love to hear from any of you if you've got any ideas for any new webinar topics that we could cover in future webinars. So please do get in touch. But once again, great to see you all here. Thank you so much for coming along. Thank you once again to Stephen. And that leaves me to say, have a good weekend when it comes. Thanks, everyone.
Thank you.